Thanks so much for having me. So my name is Ava Sullivan, like Dr. Markotter said. I'm from EcoHealth Alliance, based in New York City. I'm going to be talking today about the anthropogenic activities that influence spillover opportunities. I really want to appreciate Dr. Markotter and her entire fantastic team for inviting me here. I'm excited to sort of walk you through this topic. I also have another short slot tomorrow where I'll talk about complementary topics to this one. So today the key discussion points will be what makes this point of spillover so crucial to our global health security and also to your surveillance plans as you develop them. I want to get a little bit specific in terms of in what ways do humans drive disease emergence, and where do human activities fit into the pathway of spillover. It's been really exciting to be part of this program because we're really tackling this idea of spillover from all these different angles, and it is a complex process. So the ways that humans fit into that process is just one part of the puzzle. We'll also talk a little bit about how we can understand human risk factors for spillover and how that then translates into behavior change and points for intervention. And then tomorrow, so that's a lot of sort of theory. This is going to be high level, I guess we can call it. And then for tomorrow, we'll get into the nitty-gritty of the methodologies. So tomorrow, I'll be taking this sort of theoretical underpinning and talk about what methods we can use to understand human behavioral risk, what data needs to be collected to link exposure to outcome, and what the strengths and weaknesses of some of the tools that we use to collect this data are. So what kind of information do we gain by using qualitative, quantitative, observational methods to understand behavioral risk? So we'll start off with the challenge, and this has been framed in a couple different ways. So I think at this point I'm sort of preaching to the choir here, but really what we know are that 60% or more of emerging infectious disease events are caused by zoonotic pathogens, so non-human animal sources, and about 3/4 of those are caused by pathogens with a wildlife origin. So these are really a pressing concern and an increasing threat to global health security with increasing awareness thanks to the coronavirus pandemic. Now, the number of EID events caused by pathogens originating in wildlife are increasing over time despite our increased capacities in public health, hygiene, and medical technology. So this supports, the suggestion that these are increasing and significant to our global health. Now, what we're coming to understand about this threat is that it is not sufficient to respond to these threats after they've spilled over into human populations. So responding to outbreaks after they occur isn't sufficient based on illness that occurs at that point, deaths that occur, and the astronomical economic losses due to outbreaks in humans. So this figure here on the right is a conceptual model that really shows the value added of working at the point of spillover to combat these threats. So what we see in this light orange here are the pathogens that are circulating in the natural hosts. And those case numbers fluctuate at a predictable rate. Now then there are points of spillover that then lead to amplification and spread both in livestock populations and human populations. So really what we want to do is combat those as close to the point of spillover as possible in order to prevent outbreaks rather than respond to them. I think, one of the best examples of this is really thinking about COVID-19 and how from an epidemiological standpoint, it was perceived that we identified COVID-19 extremely close to the point of early on in the pandemic. There was a small cluster of cases that we identified, and it seemed to be perhaps an opportunity for a success story of rapid identification. But as we're looking back, we understand that amplification and spread was occurring, and that's a missed opportunity, and really is a way that rapid spread occurred. Let's see. So to really understand how to combat at the point of spillover, we have to think about spillover as a process, which inherently involves human, animal, environmental interactions, and breaking it down further to understand the mechanisms behind that. So viral mechanisms, genetic evolution, or microbiological criteria are not sufficient to fully understand risk and zoonotic potential, no matter how granular we get, because these activities and this research are about understanding the hazard, but exposure needs to occur for the spillover process to occur. So there are viruses that we're understanding. There's plausible scenarios for human exposure, for human spillover, but the exposure isn't there, and so that adverse event will not occur. So human factors tend to be extremely salient factors in driving disease emergence, and in order to understand risk of disease spillover, it's important to understand how interactions between humans and animals can influence spread. I'll just illuminate this picture a little bit for those of you who it's harder to see. This is a picture taken in the Indian context. And if you zoom in, you can really see how many different points of exposure there are between humans, animals, and their environment. There's agricultural space here, there's living quarters here, and also multiple taxa present just in this small swath of community life. So it's important to understand that the risk of zoonotic disease emergence is not uniform across countries. I'm glad we're revisiting this hotspots map. This is a building upon the hotspot map that we looked at earlier from Dr. Wood, who presented the Jones hotspot map. This is a hotspot map, Allen et al. That's a result of work from EcoHealth Alliance and others. The global hotspots and correlates of emerging zoonotic diseases, that's the title of the paper, analyzes demographic, environmental, and biological correlates of disease emergence events and creates this spatial model to describe the patterns of potential zoonotic disease emergence. The heat map illustrates predictive relative risk of zoonotic EID events, specifically estimated risk of event locations after focusing reporting bias, which Dr. Woods also described. So the results of this model suggest that disease emergence is elevated in areas of tropical forest regions, high mammalian diversity, and the regions that experience anthropogenic land use change related to agricultural practices. So this map has broad implications for surveillance, monitoring, and control of emerging infectious diseases. But just as Dr. Markotter mentioned, we have a real need to zoom in and make and dive deeper into these models and create localized and more granular spatial maps like this to understand risk. Specificity comes from adding factors that are relevant to these regions that we want to zoom into, and that's really what I'm advocating to do here, thinking about human exposure. So I'm going to describe some key upstream drivers of disease emergence. Now, these are big global drivers, and they're really at the highest point in the emergence pathway. So again, we need to add local granularity to this, and I think I'll try to walk through how to get there eventually. But the three sort of most well characterized the risk pathways that are repeated over and over again in the literature that are implicated in disease emergence are land use change, livestock production, and wildlife trade. And these are global phenomenon, but they have a few things, and they function in different ways to create circumstances under which diseases can emerge, but they do have some things in common. One is that I would assert that these are really an environmental and a human anthropogenic factors. That's the convergence of these two. So these are deeply rooted in what's going on with the environment and human behavior. There are also circumstances under which human, wildlife, and livestock species come into forced contact. And in many of these, under these scenarios, habitats are fragmented so that natural habitats become smaller. People move towards edges of forests or other pristine landscapes. And the density of viral hosts change in some way. So in the case of livestock production, we have high density of often a single species. During in wildlife trade scenarios, we often have interspecies, artificial mingling of different species. So land conversion leads to spillover as humans, reservoir hosts, and vectors become aggregated across the landscape. It can also attract wildlife, such as bats, by providing different substrates, different food, different shelters, and supporting high densities of zoonotic reservoir hosts and humans in close proximity with each other. And as these wildlife habitats are removed, reservoir hosts crowd into single areas. Livestock rearing leads to change in disease dynamics at the wildlife livestock and the livestock human interface, increasing risk of spillover. And we've seen in the case of avian influenza, livestock serve as an amplifying host to viruses that were found in natural reservoirs. And then wildlife trade, the trade of livestock, wildlife, plants, food items, and other goods can also inadvertently transport associated pathogens and introduce them to unexposed populations. So these are all high level. They have complex mechanisms folded within them, but really they have some key similarities, mostly related to that wildlife, livestock, human interface. These are some more upstream drivers that I won't speak to in as much detail, but they are also areas in which ecological and anthropogenic factors converge. I would say these are slightly less characterized in the literature as those main three that I described. They tend to have similarities between those larger ones, that is, habitat fragmentation, forced crowding, and of course, increasing that human-animal interface. But I also think that many of these, particularly population growth, agricultural intensification, and human travel and movement, are big factors in terms of amplification and spread. So once spillover occurs, these are also mechanisms by which amplification occurs. I'll also mention that these upstream drivers are not singular pathways. They work together to increase disease emergence. I think climate change is a really functional example of this. So as we're thinking about risk, climate change doesn't work in a single way to affect disease emergence, but rather it affects the hazards themselves, the exposure, and our vulnerability to disease emergence. So I'll get into that a little bit, actually. Let's talk about risk. So I just put up a quote here, which is sort of a North Star for some of my work from Emily Gurley, who does a lot of fantastic work in this space. She asserts that preventing emergence of new zoonotic viruses depends on understanding determinants for human risk. And so I want to pick apart a little bit what we refer to when we refer to risk. And I think this is important, especially if we're starting from the place of a high-level pathway, to really be able to get specific about how that functions. So risk, at its broadest sense, is hazard times exposure times vulnerability. And all the different disciplines that we're bringing today, these elements mean different things to different people. So, but most broadly, I think this is how we all can understand risk. And also, at a very broad level, we're thinking about the risk of zoonotic disease spillover events with pandemic potential, the hazard being what viruses are circulating in which animal hosts and where, and getting really specific about that hazard. Exposure is really where a lot of the work that I do lies, and that's what brings us into contact with animals. And furthermore, what contact is relevant for spillover. So of course, not all exposure is created equal, and we would have nowhere to go if exposure to animals was the answer to ending disease emergence. So thinking about relative risk related to what contact is relevant to spillover is very important. Lastly, there's vulnerability, and vulnerability modulates risk by affecting how susceptible different groups are to viral spillover. So these tend to be behavioral, social, and environmental constructs that make us susceptible to disease once it emerges. This can relate to things like healthcare, but also can relate to elements like immunity. So in thinking a little deeper about spillover, I just want to share this model because it's been extremely helpful to me in my work. I also think that it's really special to share this model with such an interdisciplinary group because it, at its core, is a model which demands a One Health approach. So this is called a Swiss cheese model. I believe it comes from the world of disaster management, which is where some of my background is from. This model is a way to understand how an adverse event occurs through complex processes. So what we can understand through this model is that there are multiple elements that have to occur and line up for an adverse event to occur. And in this case, the adverse event is spillover. At the top here are some of the dark green layers, which are related to the reservoir host. And we've heard some really fantastic talks today about just those layers. So reservoir host distribution, density, and pathogen presence within the hosts themselves. And then as we get closer down, it's about pathogen release and the ability for, the mechanistic ability for the pathogen to move from its host to another species. On the bottom, in the red, we have virological elements of spillover. So thinking about immune response. What I am working on here today is this tan layer here at human exposure. So this layer I find particularly exciting because I think that it has a high chance for modifiability and our ability to create interventions around that layer. And hopefully, if not make it a solid layer, to sort of shift things so that that spillover can occur. So we talked about this crucial point of spillover, that's sort of the when in this scenario. We talked about human exposure in the context of these high-level drivers. So I want to get even more granular and talk a little bit about behavior. So, why study behavior? The study of behavior provides context to human exposure under the pressures of those previously mentioned high-level drivers. And the solutions to plugging that hole in the layer of human exposure, in that Swiss cheese layer, is not to unilaterally end exposure to animals, right? It's to understand the elements of risk and who is truly exposed in a meaningful way, and also where that vulnerability exists. So to understand context and behaviors of communities living and working at human-animal environmental interfaces, and to create some sort of framework where we consider what is high risk for viral emergence. I provided just a few examples of behavioral, disease emergence that occurred really because of behavioral factors or in light of behavioral factors. On the on the top right corner, we have an interesting study that linked exposure of lepto at wet markets in Malaysia. So this is an occupational hazard. And occupation is a common theme in behavioral risk work because occupation is a repeated action, right? A lot of us spend a lot of time at our occupations, and also relates to it being modifiable. So if we're thinking about occupations, we're thinking about people's livelihoods, and that's a really important consideration. On the bottom here, we have studies that have come out of a case study that's repeated in our talks today about Nipah virus in Bangladesh. This is a really fascinating case study in that there's found to be higher exposure rates to folks in, particularly in Bangladesh, who drink date palm sap. Now the mechanism of exposure here is that fruit bats feed on date palms and were opportunistically drinking the date palm sap that's been collected by folks who then collect it and drink it unpasteurized. So this is a really interesting high-risk interface that couldn't have, I think, is just really highlights how much behavior and local behavior can affect these events. The third on the left is it's not a behavior that occurs at the animal-human interface, but is a behavior that occurred at the human-human interface, but changed disease dynamics in an interesting way, and underscores the importance of really gaining specificity when we think about behavior. So really what we're doing by studying behavior is zooming in to understand that local context of these anthropological drivers that I described, and thinking about the presence of hazards and how that's modulated by exposure. The idea is to work with communities and those with local knowledge to understand what interfaces exist, and then using the methodologies that I'll talk about a little bit tomorrow to describe and characterize the interfaces. This is just a modified figure from a Plowright paper, but really what it asserts is that it's not simply the pathogen interacting with the reservoir host at these high levels, but really encouraging us to go further and thinking about the exposure interface or mechanism. So in the case of Nipah, we have the natural reservoirs and hosts. We have a presumed ecological driver. We can understand routes of transmission, but then thinking really about how that exposure happens can be a little bit more dynamic. And when we think about these interfaces, this is really where we glean the information that we'll then create intervention points for. I'll also just mention, this just came to me, is that sometimes thinking about the high-level drivers and the ecological level drivers, those tend to be the best areas for policy work, whereas the exposure interfaces and the mechanisms tend to be good starting points for interventions. So we're looking towards the human-animal interface. And when we're thinking about risk, we're working to understand the context within within which risk occurs. We want to understand human contact with animals to determine which human behaviors and practices are associated with spillover. We want to be focusing on specific behaviors and practices with evidence of spillover, and we'll talk a little bit more about how to do that tomorrow. The social and economic and ecological circumstances that underlie risk behavior, and after identifying those risk behaviors, the circumstances that can be changed to prevent disease transmission. So this is sort of almost like a credo for those that are trying to, think about behavioral risk. So an example of behavioral risk work that is being done is to think about Ebola virus ecology and transmission. This is an illustration that the CDC created. And I think something that's interesting is I'm sure many of us are aware of this case study. But I only included this just to show that there's a lot of characterization that's happened at the human-to-human interface here. We have a better understanding of the human-to-human transmission, and it's better characterized even here on this basic illustration. We have 3 examples of how Ebola might transmit from human-to-human contact. But what I want to see, what I'm advocating for, is to think about why this person is here interfacing with wildlife. The reason being is that once human-to-human transmission occurs, there are different interventions that we can use to control an epidemic or an outbreak. These tend to be challenging, and we can think about it with the example of the coronavirus. If we're working on human-to-human transmission, then we're talking about masks, we're talking about limiting travel, we're talking about social distancing. But really, there's a lot of power in thinking about how we can stop that before that spillover has occurred. So that's the only reason I wanted to show this illustration today. My slides have some pictures, but maybe I'll just share them with you after if you're curious. But these bat-related interfaces are a result of thematic analysis from the PREDICT project. PREDICT collected qualitative and quantitative data that culminated in the behavioral risk products that PREDICT created, and analyzing the issues that came up within ethnographic interviews that PREDICT performed. These 5 bat-related interfaces were developed as salient to bat-to-human interfaces related to disease emergence. So we have bat guano harvesting and farming, hunted bats in the value chain, which includes bushmeat but is not exclusively so, shared food and environmental resources, bat community interfaces, which includes livestock and bats' relationship to livestock, and ecotourism. So these are just a few examples of when you really talk to communities, the types of bat-human interfaces that arise. And within these interviews, there was much more specificity that arose from here, but these are the broad bat-related interfaces that were identified. Some of the specific contexts under which these came about included bat guano harvesting in Thailand, where this is a picture that I have on my slide. Forgive me that you can't see it, but people's livelihoods are inextricably linked to the bat-human interface. So this is an area where intervention development really has to be sensitive. Another really dynamic and interesting interface is something like bat cave tourism or temples in, I think again, this is Cambodia or Thailand, where bats and tourists intermingle. Another product of PREDICT, this is a categorical output of key target demographics. The categorical outputs come from surveys that were done under PREDICT. And then these are organized across the key pathways that I described at the beginning of my presentation. These target demographics that are salient to animal-human interfaces, all of these, I think most of these are occupational. But I think something that's really exciting about working with social science methods and also working at a community level is that you understand that this goes beyond simply occupational risk. Especially in the case of occupations, the deeper you dive, the more you realize that this actually doesn't stop at just being related to their occupational, their occupation, but rather gender, social economic status, their many other elements of their life. So, for example, some of these demographics are exclusively put onto females in communities, or excluded by certain religious practices, for example. So when you get really granular about these different interfaces, it changes the opportunity to create interventions around them, and it changes our understanding of who's actually at risk. So I'm going to end here to really just describe or reiterate the goals of behavioral risk characterization. And this is where we're going to pick up tomorrow when we talk about methods. So I hope that I didn't get too much into the theoretical here, but if you have questions about how we turn this theory or these concepts into practice, make sure you write them down because I'd love to answer questions about it. Really what we want to do with this information is to identify and monitor behaviors, attitudes, practices, and norms that facilitate human-animal interface and influence spillover. Spillover most importantly, but also amplification and spread of zoonotic disease. The second is we want to be able to combine that data and link exposure to outcome in context. So I think this is really where some of the stuff starts to get exciting, is where we can link some of the qualitative and quantitative information about communities and link them to what's happening in bats, what's happening in livestock, what's happening in the environment, and place it into context. And three, we want to take that information, that rich information, that mixed method output, and target high-risk behaviors for intervention and or policy recommendations. All right, and with that, I am going to end there, and really, please reach out or find me afterwards if you have questions or things you want to hear for tomorrow's talk. Thanks.